Hi everyone, good afternoon to all. Today we are here with a lecture of the cell. As we know, the cell is a structural and the basic unit of life, as it is structural as well as the functional unit of a living being. So today we are going to study with the topic cell and its organelles. Mainly our focus will be on a fluid mosaic model, perioxysomes and their cell membranes transport system, how the transport system operates in a cell membrane. That is, as we know, the cell membrane is a semi permeable membrane. So cell, it was discovered by the Robert Hooke, as he observed the slices of the cork cells. Okay, all right. And the outermost boundary is our cell membrane. Mark my words, this is a typical diagram of a animal cell. I am representing an animal cell. This is a cell membrane and this is our nucleus. This is nuclear membrane. Okay, this is our DNA material, that is chromatin. Okay, here is the endoplasmic reticulum. Here is the endoplasmic reticulum, short is ER. Okay, and this endoplasmic reticulum, ribosomes are present over it, so this is basically rough endoplasmic reticulum. Okay, because it contains the ribosomes. And here is a sausage shaped structure known as mitochondria. Okay, and the dense fluid inside the cell membrane, which separates, as the cell membrane separates the extracellular fluid and intracellular fluid, the liquid or the dense fluid inside a cell is known as the cytoplasm or cytosol, you can say. So I'll mention it here only, cytosol. Okay, cytoplasm or cytosol, and these are Golgi apparatus. They were discovered by the Camillo Golgi and they are used for the packaging of materials. Okay, the main function of endoplasmic reticulum, firstly these are of two basic types: endoplasmic reticulum, the rough endoplasmic reticulum, or the smooth endoplasmic reticulum. The rough endoplasmic reticulum contains the ribosomes, and ribosomes, as we know, are factories of the protein synthesis. So the main purpose of endoplasmic reticulum is to synthesize the proteins. They will going to synthesize the proteins. And smooth endoplasmic reticulum, the function of the endoplasmic reticulum means without ribosomes, lipids and steroid synthesis basically takes place in a smooth endoplasmic reticulum. Now we are going to have the details of all these organelles. First of all, as we know that we were talking about the RER and SER, which are the types of endoplasmic reticulum. As I told you earlier, the rough endoplasmic reticulum contains the ribosomes. This structure contains the ribosomes, and as we know that ribosomes are the factories of protein. So the main function of RER is to synthesize the proteins. Clear? And SER, smooth endoplasmic reticulum. Smooth endoplasmic reticulum. They are called smooth because they are without ribosomes. They are without ribosomes. So therefore their main function is to synthesize the lipids and steroids. Clear? Actually they are flattened structures like this. They are flattened structures. We can show it like this. This is a structure of smooth endoplasmic reticulum, and if we add the ribosomes on it, then it will become the structure of endoplasmic, that is rough endoplasmic reticulum. Coming on to our cell membrane. This is our main topic. Cell membrane. Cell membrane is the outermost boundary of a cell. Outermost boundary of cells. Its function, its purpose is to separate ICF and ECF. This is intracellular fluid, and this is extracellular fluid. Okay. So basically cell membrane is made up of lipid bilayer. Lipid bilayer was given by Davison and Danelli. Clear? Cell membrane is made up of lipid bilayer. I will show you the structure of the lipid bilayer. Okay, lipid bilayer was given by the Davison and Danelli. These were the two scientists who gave the lipid bilayer. Now the question arises how it looks like, how the cell membrane or plasma membrane looks like. There was a famous model, the fluid Mosaic model. And this model was given by Singer and Nicholson in year 1975. Okay, this model is the most important model, so we will pay attention to it. Fluid Mosaic model. It was given by Singer and Nicholson in the year 1975. Fluid which can flow, anything which can flow are called fluids. Mosaic is appearance. Likewise, you can say as if water is running and ice is floating over it. Yeah, this is a mosaic appearance. Now we will see how the mosaic appearance resembles our fluid mosaic model. Our lipid bilayer structure, how is it similar with the mosaic appearance? Okay, it was just an example. So, lipid bilayer. This is hydrophobic hydrophilic head and hydrophobic tail. ठीक है. Now I will show you the diagram of the fluid mosaic model. ठीक है. And one thing more, the fluid mosaic model, it contains basically the cell membrane, which is a phospholipid bilayer. It contains proteins 55% and lipids 40%, as well as carbs, which is 5%, you can say, or less than 5%. Okay, proteins 55%, lipids 40%, and carbs 5%. This is how it looks like. Okay, this is the arrangement of the lipid bilayer. Hydrophobic tail and hydrophilic head. Okay, these are the proteins known as transmembrane protein or the interstitial protein. These both are proteins. This is lipid bilayer arrangement, and this is the central layer. And its geometry is like this. Carbs are arranged over here. These are proteins. Okay, and in the proteins, these are chains of protein known as glycoproteins. Okay, and cholesterol is present over here. Cholesterol. This is a chain of the glucose molecules. Glycolipid. This chain of proteins is inside a protein, inside a protein, and is known as glycoproteins. Okay? These carbs are arranged in a chain outside a protein, therefore they are known as glycolipids. They are glycolipids and these are the glycoproteins. This is the structure. Hydrophilic head towards outside and hydrophobic tail towards the inside. Now, how it's fluid? The question arises, why fluid? What is a means of being fluid? Fluid. Fluidity. Basically, fluidity depends upon the two reasons: unsaturated fats and cholesterol cholesterol content. Unsaturated fats. Cholesterol content prevents the close packing of carbs, proteins. Therefore, molecules are freely movable, hence show movement. Unsaturated fats. Unsaturated fats are fats which are liquid at room temperature. I will write liquid at room temperature. They are liquid at room temperature, as well as they contains the double bond. Okay. These are the two basic reasons which provides the fluidity to our phospholipid bilayer. Now, remember about the fluidity. The flip-flop movement is possible. Flip-flop movement is not possible while lateral movement is possible. Wait. Suppose this is A, this is A, this is A, this is B, this is B, this is B. They cannot interchange. A and B cannot interchange their positions. Means no flip-flop movement is possible, while A and A or B can exchange their position. Therefore, the lateral movement is possible. We can say like this: lateral movement is possible and flip-flop movement is not possible. One thing more: proteins found in cell membrane acts as carrier protein, receptors. carrier protein and receptors. I hope the fluid mosaic model is clear to you, and now we are going to start with our perioxysomes. Perioxysomes, they are the organelles. They are vesicles found in our cells. They are actually they are organelle, part of suborganelle in cells. They are vesicles in which the oxidation of very long chain fatty acids takes place. VLFC, very long chain fatty acids. This is the important function of perioxysomes. They are vesicles like, and their main function is to perform the oxidation of the very long chain fatty acids. Moreover, they help in degradation of hydrogen peroxide with an enzyme known as hydrogen peroxidase. Okay, this is a catalyze. This is important function of the vesicle. One topic is more linked with the perioxysomes, and that is PBD. PBD means perioxysomal biogenesis disorder. The defects in perioxysomes which leads to the various disorders. They are under the PBD, perioxysomal biogenesis disorder. And in this, our main concern is with Zellweger's syndrome. As we have already discussed about the perioxysome, the main function of perioxysome is to deal with the oxidation of the very long chain fatty acids. How it will affect if it will not perform its function? In case very long chain fatty acids do not oxidize due to some defect in the perioxysomes, very long chain fatty acids do not oxidize. So it will accumulate in brain, kidneys, as well as in liver. Very long chain fatty acids do not accumulate in brain, kidneys or liver. That's why this disease is known as cerebrovascular disease also. Sorry. Now, due to the accumulation in the brain, kidneys and liver, it will impact the effect of the brain, impairment of brain and kidney plus liver. they will result into dysfunction. Okay, this is an overview of Zellweger's syndrome. Okay, once again I will summarize. Zellweger syndrome. Peroxisomes. Peroxisomes main function is is that it will oxidize the very long chain fatty acids. So if somehow they are failed to oxidize the very long chain fatty acids, they will not oxidize the very long chain fatty acids, and they will accumulate in brain, kidneys and liver. As a result, impairment of brain, kidney and liver will take place. Okay, and it's characterized by they are characterized by empty peroxisomes. I hope it will be clear to you. Disease number 2. Infantile refsums disease. This disease is also related with PBD, paroxysmal biogenesis disorder. Okay, infantile refsums disease. No need to go into detail. Depth is not needed. Just a name is written over here. Infantile refsums disease. I think that's sufficient for the peroxisomes. Let's have an overview of mitochondria. Mitochondria. As we know, the energy currency of our cell. Basically, mitochondria is a sausage shaped organelle. Mitochondria, as we know, is a double membrane structure, double membranous structure. We will study its structure. This is the structure of mitochondria. Outer membrane, inner membrane, cristae. Inner membrane is projected towards the inward, so these structures are known as cristae. And the space between the two membranes is known as membranous space. The space is very important so that electron transport can run efficiently, because on this inner membrane, the whole mechanism of electron transport over here, and there are F1 and F0 particles. Electron transport chain, fifth complex, ATPase, ATP synthase activates. Electron transport's final step took place in the fifth complex, ATPase synthase, and F1 and F0 particles are over it. Right? 1 means it discovered firstly. ठीक है? They both are responsible for the oxidation, for the synthesis of ATP basically. ATP is synthesis. ठीक है? This is the space, intermembrane space, which is very important so that ETC runs efficiently. In ETC, there is a fifth complex known as ATP synthase, and F1 and F0 particles are present over it, which helps in the ATP synthesis. 1 because it means it was discovered firstly. Transport. Transport is our major topic. Transport is basically of 3 types. Passive, active, as well as vesicular transport. ठीक है? Passive transport, no energy is needed. No energy is needed. Active transport, energy is needed. And vesicular transport. Vesicular transport means transport of macromolecules. Transport of macromolecules is via vesicular transport. Macromolecules, molecules, basically the biomolecules. Biomolecules having the size more than 1000 Dalton. The unit is Dalton. Clear? First of all, we will start with the passive transport. There are 2 types in a passive transport. Firstly, we will start with the simple diffusion. Basically, the solute moves from its higher concentration to the lower concentration. Solute moves from its higher concentration to the lower concentration without the use of any energy. There is no need of energy and there is no need of the medium also, carrier protein. No carrier protein and no energy is needed in a simple diffusion. It will simply moves, the solute will simply moves from its higher concentration to the lower concentration via lipid bilayer. Lipid bilayer. Examples are the carbon dioxide as well as the urea. It will simply cross the lipid bilayer solute from the its higher concentration to the lower concentration. Okay. This was about the simple diffusion. And simple diffusion, it will down the concentration gradient. Okay. This was the first type of passive transport. Passive transport is basically, in passive transport we do not need energy. Energy is not needed. Energy is not needed. Second type of the passive transport, that is second type of passive transport is facilitated diffusion. In this, of course, the energy will not be needed in this. Carrier protein is needed in it. Carrier protein is needed in it. Basically, it will also move from the solutes will also move from its higher concentration to its lower concentration, but the difference is carrier protein will be needed to have the facilitated diffusion. Simple diffusion, second type is left. Simple diffusion, the type 1 was already started. That was, it will pass through the lipid biomembrane because they are lipid soluble. Okay, because they are lipid soluble, that's why they will pass from the lipid biomembrane as it is, from its higher concentration to the lower concentration. And second type of the simple diffusion, that is, simple diffusion itself is a part of the passive transport. Alright, passive transport, simple diffusion's further type, that is ions channels through the transmembrane protein. It will take place through the transmembrane protein. Voltage gated. Example, sodium, potassium and calcium. So voltage gated, sodium, potassium and calcium, these were the example. And on the other side, ligand gated. Ligand gated, basically example is the acetylcholine. Receptors are present on post synaptic vesicles. Okay, ions channel will take place through a transmembrane protein. Okay, passive transport, simple diffusion. Simple diffusion's next type is ions channel, voltage gated and ligand gated. Voltage gated, a specific voltage is required so that voltage gated channels will be open. Examples are the sodium, potassium and calcium. These voltage gated channels are used in the signals of brain. For example, when the signal travels through the neurons, there is a process. Here is the unpolarized state, here is the polarized state, and this is repolarized state. Okay? When the neurons travels the signal, it will be travel like this, and these channels are present over here. Especially the sodium potassium channels are present over here, so that it may help the neurons to travel the signals. They are important. These voltage gated channels are important. Ligand gated acetylcholine receptors present on post synaptic vesicles. The space between the two neurons is synapse. The space between two neurons is synapse. These ligand gated channels here are the present of synapse. These spaces here is the presence of our acetylcholine receptors. Okay? When the acetyl will be released, it will come over here. Receptors are present here. Okay? These are neurons. Active transport. Energy is required in active transport. Active transport is of two types: primary active transport secondary active transport. In primary active transport, direct energy is needed. Secondary active transport. Examples of these are the sodium potassium pump, SGLT 1. We will have the deep explanation of this secondary active transport with the help of a diagram. For the better understanding of the transport, I'm going to draw a simple diagram. Here I'm drawing the surface of intestinal cell basically. We will understand the transport with the help of this, and this is our A3, you can say. Okay, this is A3. Sodium and glucose getting in with the help of a carrier protein. carrier protein, sodium and potassium going inside a cell with the help of a carrier protein. This is SGLT1. Okay. Now fructose is going inside. Fructose is going inside with the help of carrier protein. Fructose is going inside with the help of carrier protein. This is basically the example of the facilitated diffusion. Facilitated diffusion. Okay. No energy is required, only carrier protein is required. Here the energy is required, so this is a part of the active transport. This is a part of active transport, basically the secondary active transport. And SGLT is the one of the examples of the secondary active transport. Okay, carrier protein is required. Carrier protein is required. With the help of the carrier protein, sodium and the glucose gets inside the cell. They both moves simultaneously inside the cell, and this transport is also known as co-transport. Okay, this is basically an example of co-transport, or you can say the sim transport. Both the sodium and glucose are moving inside the membrane with the help of a carrier protein. Energy is required, ATP is required over here, so this is a part of the secondary active transport. Because ATP is required, they both are going inside simultaneously. This is the process of co-transport. Okay, SGLT1. Okay, the phenomena is SGLT1. This is under secondary transport, secondary active transport. Now this is an intestinal cell. Here is our pump. This is our sodium potassium pump. Sodium potassium. Again, sodium potassium pump is an example of secondary active transport. 3 sodium will move out and 2 potassium will move inwards in a sodium potassium pump. This is a pump, therefore the energy is required, ATP is needed over here, so therefore it is a part of a active transport. Energy is needed, carrier protein is needed. Energy is needed over here, and 3 sodium ions moves outward, while 2 potassium ions moves inward. Okay, this is a pump, sodium potassium pump, as an example of the secondary active transport. Okay, all right. And glucose moves outside. Glucose moves outside with the help of the GLUT2. Okay, glucose moves outside the cell with the help of the GLUT2, which is a carrier protein, helps in the transportation of the glucose. Okay, secondary active transport. There is one question there is one question which will be solved with the help of this diagram. Okay? And that question is, first of all, I'll write the question. Why glucose is added in oral rehydration solution, ORS? Why glucose is added in ORS? This is the question. So, coming on to the diagram, find its answer. And the answer is symport. Symport. Here is the symport. Co-transport or symport. How it works? In oral rehydration solution, we add the glucose because with the help of glucose, the amount of glucose added into the cell will simultaneously add the electrolytes, will add the sodium ions. Okay? Glucose is going inside the cell, and sodium is accompanying the glucose. Therefore, glucose is added in a oral rehydration solution. The symport will take place. Both glucose and sodium simultaneously moves inward the cell. Therefore, glucose is added so to replenish the electrolyte. Reason is symport. Sodium will go plus glucose with the help of glucose. Glucose will take the sodium into the cell, and the mechanism and the method is the symport. Okay? This is a transport. Normally I'm writing symport. Okay? They will move simultaneously, and the electrolyte moves inside the cell. Electrolyte moves inside the cell. Electrolyte moves inside the and replenish the electrolyte. Glucose. Glucose moves inside the cell and replenish the electrolyte. Simple transport, and symport is also known as co-transport. Vesicular transport. Transport of macromolecules these are of two types. Endocytosis, exocytosis. Endo means in, exo means out. Let's see the exocytosis. Here is the membrane. For example, plasma membrane is here. These are the vesicles. These are the hormones. For example, the hormone is PTH. Vesicle. Now this vesicle with fuses with plasma membrane in order to release the hormone in the outside environment. Hormone. This is how it works. Exocytosis. Plasma membrane, vesicles, hormones inside it. Plasma membrane, vesicles fuses with the plasma membrane and the hormones releases outward in the body. This is a process of exocytosis. Endocytosis means inward transport. These are the clathrin pits. Clathrin is a protein. Inward transport of material, you can say, of any substance. Okay? For example, say LDL. Low density lipids, LDL. Okay? It will show like engulfment like process is there. Engulfment like process takes place. Okay. So coming on to our last topic, that is osmosis. Now this is something important. The movement of solvent towards the higher concentration. The movement of solvent towards the higher concentration of solute through SPM, that is selectively permeable membrane or plasma membrane. The movement of solvent towards the higher concentration of the solute through the SPM. For example, this is solvent, this is solute, this is solvent. liquid or solvent will move. Liquid or solvent will move in with higher concentration of solute, where the higher concentration of solute is present. Osmosis. This was all about for today. We will see you soon. Bye bye. Take care. Jai Hind. Jai Bharat. That's all.